Hi, and welcome to Cloud on Air, live webinars from Google Cloud. We're hosting webinars every Tuesday. My name is Jonathan Sheffy. I'm the product manager for genomics and life sciences at Google Cloud. And today we're going to be talking about powering genomics research on Google Cloud Platform. You can ask questions anytime on the platform, and we have Googlers on standby to answer them. Let's get started. So a few topics we're going to talk about today. First, I'm going to tell you a little bit about how you can accomplish genomic and clinical data mining together using BigQuery. Then I'll tell you a little bit about how you can run batch workflows at scale using Pipelines API. Then I'll hand it over to my friend Geraldine from the Broad Institute to tell you a bit about GATK4 and the FireCloud platform. She'll also tell you about how their team optimized GATK4 for Google Cloud. And then Keith from Google will tell you a bit about some of the really cool innovations that are happening right here at GCP. But first, I want to tell you a little bit about the team that I'm a part of. This is a team that started at Google just last year. It's called the Cloud for Health and Life Sciences team. We've brought together product managers, software engineers, solution architects, and a commercial team to help organizations do all sorts of amazing things with health and life sciences data using Google Cloud. Whether it's an academic institution, whether it's a pharma company, a provider, a payer, a health products or services company, we want to help you use any kind of health and life science data type in Google Cloud more effectively. In particular, the product teams that I'm a part of are focused on a few key modalities. Clinical data, so EMR data, EHR data, other kinds of phenotypic data sets. Imaging data, that's DICOM, PACS, PET scans, MRIs, X-rays. And my team, the Genomics and Life Sciences team, where we're dealing with not only genomic data, but all kinds of other biomedical data sets. So could be microbiome, RNA-seq, and others, flow cytometry. Why we're doing this is because our vision is that there's going to be a convergence of these data types in the cloud, where for whatever sample you're working with, whether it's yeast or an entire human population, it'll be helpful for you to be able to analyze data across these different modalities together. And that's what we're enabling. So let's talk a bit about what that can look like, especially coming from the genomics and life sciences point of view. So one of the products from my team is called Variant Transforms. Variant Transforms is an open source tool to help you manage your genomic data using BigQuery. You can think about it as a way to take your VCFs from panels, exomes, or genomes and import them directly into BigQuery using the inherent VCF schema you're presenting. From there, you can do things like calculate your allele frequency, or build a cohort, or do ad hoc exploration of that data set. But I think we all know that where these kinds of data sets get really exciting is when we join them with other kinds of data. So that's exactly what a company called Color in the Bay Area did. And I want to tell you a little bit more about that. So Color is a health services company. They offer very affordable and accessible genetic testing. And they have a 30 gene panel in cancer. And they also had an existing database of phenotypic data, some health history data, and report data that was sitting in a Postgres database. The problem they had is that they wanted to be able to expand that test by mining the raw sequence data alongside the other data they had available on those samples. The cloud provider they'd been using up till now didn't really have a mature enough managed data warehouse solution like BigQuery, which is so great for these kinds of scalable queries. So what they do? They used Variant Transforms, as I mentioned, a totally open source tool for bringing all of that data into BigQuery. They took the data from the Postgres database, which had a bunch of biomarker information, and put that in BigQuery and joined it together. On top of that, their data scientists stood up all kinds of really cool machine learning tools and built a whole bunch of great models. So what happened? Well, they got all the feature engineering done in 1 week, and they created 3 novel models that outperformed an existing major clinical model for predicting certain biomarkers. In fact, their test, which is actually designed for cancer, they found out worked in a completely unrelated therapeutic area. This is really exciting. It may become a publication, may become a novel test, but we're really excited about the kinds of results that our customers are achieving by joining their genomic data with other data types using BigQuery. So I'm going to switch gears for a minute here from tertiary analysis to secondary analysis. No doubt if you're watching this, you're probably processing lots of samples, perhaps in a cloud environment, perhaps somewhere else, and you're thinking about how you can really scale up that work of your pipelines. So our team also offers a API called, not surprisingly, Pipelines API. Pipelines API helps you run bioinformatics pipelines at scale, in achieving great metrics around turnaround time and cost. You can stand up all kinds of great industry standard tools like GATK, which you're going to hear more about, and you can optimize for parallel execution, and you can leverage a lot of great innovations in Google Cloud, like preemptible virtual machines, GPUs, regional storage. You're going to hear a little bit more about that later today. We've also brought together a phenomenal ecosystem of platform partners and workflow engines to help you with all this. So soon you're going to hear more about Broad Institute and their FireCloud platform. And you'll also hear about the Cromwell open source workflow engine, which are great ways of running your workflows and leveraging the power of Pipelines API. We also have other partners such as Wuji Nextcode, DNA Stack, 7 Bridges, and the Nextflow open source workflow engine. The kinds of results that people get from leveraging Pipelines API is this kind of massive scale. So for example, the team at Stanford School of Medicine is be able to run thousands of genomes in parallel, what today they say feels like infinite resources, and be able to boot up a lot of machines at once to accomplish a batch workflow in a very short period of time while still focusing on low cost. Our friends at the Broad have used Pipelines API quite a bit, shall we say. They process over 250 genomes every single day. They are now working with over 35 petabytes of data in Google Cloud, and just last year consumed over 100 million compute core hours. And they're scaling up their work because of the innovation within Pipelines API. So let's talk a little bit about some of the work that they're doing. The Broad gnomAD team recently called variants on the gnomAD dataset with the GRCH38 reference. And the problem they had was that gnomAD, as you might know, 79,000 whole genomes that were previously called with HG19. They wanted to be able to call variants for that entire data set using GATK4 and GRCH38. So they were able to spin up 79,000 workflows and call the variants for the entire data set in 3 days. It's a really exciting result, and I think is a great example of the kind of scale you can achieve using Pipelines API and engines such as Cromwell and platforms such as FireCloud. So what's next for that team? They're going to be calling variants for exome data on gnomAD as well, again, using some of the same technologies and techniques. So speaking of innovations in Pipelines API, just last month we released a brand new version of Pipelines API for our ecosystem of platform partners and workflow engines. What's new? Well, first, you're able to run multiple Docker containers per instance. This is really helpful because you can now run service containers in the background while a workflow is running in progress. We also make it a lot easier for tool authors to be able to inject their own containers around a workflow. Localization and delocalization of files is much, much simpler. It's just now a container instruction. And there's also, probably my favorite feature, a lot more information is available to our platform partners and workflow engine partners on the information about the transition between states. So that and on failure modes. So for example, when I have a pipeline that fails, why did it fail? Did it run out of memory? Did it simply crash? Did something happen to the node somehow? Did I get preempted? That information was a little harder to access before, and now it's much easier and provides a better experience for whatever workflow engine or platform you're using. We've also made it easier to specify regions in zones, and much easier to specify minimum CPU platform options. Keith will tell you a little bit more about that soon in terms of how you can bring up custom VMs. So all this great talk about secondary analysis, let's learn a little bit more about how people are doing this in the real world. So now I'm going to invite up my friend Geraldine from the Broad to tell you more about GATK4 and FireCloud.
Thanks, Jonathan. It's a pleasure to be here. I'm going to talk today about GATK4 and FireCloud, and the way we have been working to make our pipelines available, accessible to the wider community of researchers. Now, first, I want to start with a little bit of a recap of what is GATK exactly, what that covers. So GATK is a genomic analysis toolkit, and this is roughly the process for genomic analysis, specifically with a focus on variant discovery. If you think that you're starting with biological material, for example, blood samples, we're going to extract DNA from that, put that through the sequencing machine, right? That's going to produce a huge pile of short reads. Those short reads, that's the raw data that we're going to be using in our pipelines. The end result that we want to get to is a list of variation, mutations in the individual genome or genomes of a group of individuals. And so the pipeline, which is what GATK constitutes, is going to take that raw data, do some cleanup, do some mapping to find out where the reads belong in the genome, and then do the analysis that enables us to identify those variations, those mutations in individual genomes, which is ultimately what the researcher cares about. Now, these tools have been around for a long time, about a decade, which honestly in genomics time is pretty much like a century. And for a long time, they were evolving kind of naturally. We're just improving our ability to just push the scientific boundaries of what we could identify. Now, over the last few years, as Jonathan mentioned, there's been a huge explosion in the size of the genomic data and the data sets that we need to be able to analyze. And so we've put a lot of work into making sure that the tools and the pipelines can cope with that huge explosion in the size of data sets. And specifically, we built what we call GATK4, which is version 4 of the GATK toolkit. I'm not going to go into a detailed laundry list of features, but the main themes are that we've really improved the technical performance, the speed and scalability of the tools. And that has involved a complete rewrite. We've rewritten that entire software code base from scratch to be able to cope with the demands of the genomic workloads that are now today's reality. But it's not just about the technical performance, although that's very important. But we've also put a lot of work into expanding the scope of analysis to be able to cover more variant types. So where previously we were very focused on germline short variants, that was kind of what we were known for. Now we've expanded that to cover all the major variant classes, including copy number, structural variation in both germline and somatic mutations. So we're now able to cover almost all of these use cases, and the remaining one that is not quite implemented today, somatic structural variation, is planned. We have that on the roadmap. We hope to deliver that later this year. And so the idea is that you have all these pipelines that allow you to really do all the scientific work that you need related to variant discovery. Now, the thing is, and there's been two big pushes in that direction. One is just developing the right methods to produce those end results, but also improve the speed and performance of the tools, right? And that has involved leveraging a lot of the improvements in, for example, machine learning that have really flourished over the past few years. Now, if we look at these pipelines, all of them involve a sizable number of steps involving different tools. And for the individual researchers, it's not easy to implement this at scale. And so one of the things we've been focused on lately is formalizing pipeline definitions, which we call the GATK best practices, which a researcher can take and apply to their data without having to figure out by themselves how to implement this. So we call that the GTK best practices pipelines. But there's still a big step between having the pipeline definitions and being able to implement that. And so we've gone a step further, and now we're making available these pipelines as ready-to-run pipelines that are preloaded in a platform that we call FireCloud, which runs on Google Cloud. And so we have these workspaces that contain the pipeline as well as example data. And so anyone can go and clone one of those workspaces, run the pipeline as it is pre-configured on the example data. They can also upload their own data and run the pipeline on their own data. And that way you have the GTK best practices pipelines just available in a ready-to-run state out of the box. So hopefully I've got you interested. And I want to tell you a little bit more about FireCloud's platform itself. Okay, so FireCloud is meant to be a self-service analysis portal that anybody can access, and specifically anybody can access for free. So we built this originally for broad researchers, but we make this available for free. Of course, it's using Google Cloud resources, so you still have to pay Google Cloud for storage and compute costs, things like that. But we do have a free credits program that you can use to get started with the platform to test it out without taking any financial risk yourself. Now, the platform itself offers a number of services, including the ability to manage your data, to share your data securely, and to run workflows on your data. So we've got a number of pipelines that are preset that you can run on your data in this platform, but you can also bring your own pipelines, either variations of ours or just different workloads that you need to run, because we understand that many people use other tools besides GATK. And we can use this through the web portal, which is a graphical user interface. You can also use it through the API if you prefer to interact programmatically with the platform. And as of last week, we also have now the ability to spin up Jupyter notebooks on Spark clusters. So you can follow up your batch execution, your pipeline execution, with interactive analysis of the results that they produce. Now, let's look a little bit more closely at how batch execution of workflows works in FireCloud. Don't worry about the details yet, but let's look at the overall system. Here we have two researchers who have access to this workspace. The workspace encapsulates everything that's involved in a particular project, including the definition of the data that's involved and the methods. And that's also the place where you're going to be executing pipelines. So you also have access to pipelines, workflows, that's a stack of scripts that you see above the workspace. And you have a number of data buckets that are available. Now, the way it works is that the data is stored in buckets on Google Cloud. So your data is going to live in Google storage, either associated with the workspace or in other buckets. And you can also compute on data that's elsewhere in the data library, which are public data sets that are provided. The workflows are WDL scripts. WDL is a workflow definition language. The GATK pipelines are all provided as preloaded WDL scripts. And that contains the definition of your actual workflows. Now, when you go to run a workflow on your data, all you need to do really is connect the workflow with the inputs. And the inputs, that's the data that's defined in your data model, which describes what files you have available and how they plug into the workflow. So you have your workflow, you have your data definition. You're going to launch one of these workflows on your data. That's going to be handed off on the back end to Cromwell, which is the execution engine. And Cromwell is going to take care of everything for you. So it's going to execute your workflow, talk to the pipelines API, which Jonathan talked about, and run your workflow on the Google Compute Engine, and then write back the outputs to your workspace data bucket. So that's how you run a pipeline. in FireCloud. And specifically, if you're using one of the GATK best practices pipeline, which is preloaded, you can get started with this very quickly. And it abstracts away all the difficulties of implementing these kinds of complicated pipelines on the cloud. And what's really neat is, like I said, as of last week, you can follow up and use the results that you have. Now you can explore your results interactively in Jupyter Notebooks. All right, so that shows you how to access and run the GATK pipelines in FireCloud. Let's talk a little bit about what it costs, because when we started migrating our workflows to the Google platform, it became clear to us that there were some great opportunities to cut down the cost of operation. And here I'm going to talk briefly about the various strategies that we used to bring down dramatically the cost of operating our genome analysis pipeline. Now, the main strategies that we used, and we developed this with quite a lot of help from our friends at Google, have been in several different categories. One was this idea of task splitting and persistent disk. Now the idea here is that when you have your tasks in a workflow, you're allocating for each task, you're allocating some resources, some disk size, some processor size. And the beefier the machine, the bigger the disk, the more you're going to pay for this hardware. And so what you want to do is make sure that the allocation is commensurate with the needs of your task. And we have found that there are various ways that we can optimize how tasks are either split up or bundled together, depending on what their computational requirements are, and what is the size of the inputs and outputs that we need to store locally, to really optimize the allocation resource requests. Now, the other, and that kind of goes with it, the other big strategy, and this is personally my favorite, that we've applied to bring down the costs, is the use of preemptible VMs. Preemptible VMs are machines where you get them at a really steep discount. You pay 20% of the normal price for a machine. But that comes with the understanding that you can get kicked off the machine at any time. So for really long-running jobs, that's not so desirable. But if you're running really short jobs, or up to a few hours, it's actually a really good deal because you're paying a lot less for the compute. And there's some logic built in so that if you do get kicked off the machine, there will be some automatic retries. And you can even tune how many attempts you want to do on preemptibles before defaulting back to regular machine that you have for short. And so by doing that and by switching virtually all the steps in our pipelines to using preemptible VMs, we've gained an additional 35% savings on the cost of running our genome pipeline. And now the final piece in the puzzle, the last big strategy that we've applied to cut down costs of our pipeline operation is to be really smart about how we're accessing the data. JP Martin at Verily Life Science contributed some really valuable code to HTS GATK, which is an open source library that underlies GATK. It's what GATK uses to read in and write out files. And that's the code that JP contributed allows us to read data directly from the bucket instead of having to copy the file to the local disk before we can compute on it. And that makes a big difference because, for example, if we're parallelizing across genomic regions, if you think that normally you would have to copy an entire genome BAM file to local disk, right? But now you can skip that and just copy the little piece of data that you actually want to run on. That allows you to go much faster because you don't have to wait for 100 gig BAM file to get copied. And it also allows you to allocate a much smaller disk space. So that ties into the first strategy that I mentioned, where we can use much smaller disks for each task. And if you're thinking, well, this is going to be used across thousands or tens of thousands of genomic intervals, and then replicated across tens of thousands of genomes themselves, that amounts to fairly significant savings. Again, in this case, 15% savings on the total cost of the pipeline. And the result of all of that in aggregate is that from an initial cost of $45 per genome to process an entire genome on Google Cloud, we've cut that down to $5 per genome. And we've done some additional tweaking that I can share. We're seeing now that with some of those additional tweaks, we can actually bring down the cost of the pipeline right now to $3.65 per genome. So that just gives us so much more bang for our research buck. It has made a huge difference in terms of what we can do at the Broad Institute with our genomic pipelines. I want to make clear that we're still offering best practices pipelines that are generic, that can run anywhere, but we have this version of the GTK4 pipelines that is very optimized, very deeply optimized for Google Cloud. It's what we run in production, but we make that available so that everybody can benefit from this optimizing work. You can try this pipeline in FireCloud. It's preloaded in the workspace that lives at the URL that's on your screen, the $5 genome analysis pipeline, which we might have to update the name of to the $3 genome pipeline, and hopefully that will just keep going down. So that covers the work that we've done so far with GTK and FireCloud. I'm going to throw it over to my friend Keith from Google, who's going to talk about platform innovation.
Great. Thanks, Geraldine. So innovation is something that we hold dearly at Google. And as you heard from Jonathan and Geraldine, our innovation stems from things like the Pipelines API to BigQuery, even to stuff like Preemptible VMs. It's fascinating to think that for the price of a cup of coffee in New York, you can do an entire genome analysis. To me, that's amazing. But our innovation doesn't just stop there. At Google, we don't believe you should simply lift your workload or application into the cloud. We believe that you should lift, shift, and improve upon that workload and application. And the improvements take form of some of the cost innovation and technical innovation that we have. For instance, we have a live migration capability. So your instances can seamlessly migrate to a machine during maintenance windows without any user intervention and without any planned downtime. This is something we took advantage of during the recent Spectre meltdown vulnerabilities, whereby we migrated our entire customer environment seamlessly without them having to be involved in order to address that vulnerability. Additionally, we have custom machine types. So we know that applications sometimes don't fall into a specific ratio of CPU and memory. So we allow you to define what that ratio is. Maybe you need 4 cores and 80 gig of RAM. We don't stop you from doing that. Additionally, we have recommendations using our vast history of machine learning to look at how an instance is performing and say, you know what? If you change the core count on this machine, or maybe reduce the memory footprint not only will your instance still run, but you'll save a couple dollars as well. We believe that you should pay for what you consume. And to that end, we have per second billing. So if your instance runs for 73 seconds, you're going to pay for 73 seconds. You're not going to pay for 120 seconds or even more. We also have extremely fast startups for our instances. We're talking about a matter of seconds. So this gets you up and running as fast as possible. And lastly, we allow you to resize your disks on the fly. If you need a bigger disk, we allow you to do that seamlessly, as well as increasing the IOPS for that disk as you make it bigger. Now, innovation isn't only about technology and cost. We think security is also a strategic part of innovation at Google Cloud. And it starts with you, the customer. By default, without you having to do anything, all of your data in transit and at rest is encrypted. You don't have to worry about any PKI infrastructure. You don't have to worry about any key management. Now, of course, if you are a type that likes to manage your own keys, we allow you to do that. But knowing that you don't have to do that is an extremely important benefit. And this notion of security and encryption extends into our cloud stack. It starts at the silicon level. We made our own in-house secure chip that we put on every peripheral device, and that secure chip will check to make sure that it's a trusted root during boot. If it detects any abnormality during the manufacturing cycle or even during steady state, that machine in our data center will not boot and will be reported to our engineers. That extends into our server environment. We have our own custom servers that we manufacture, and having the ability to make our own servers, we eliminate all the extraneous stuff that you may have in a server from off the shelf. We don't necessarily have to put in video cards if they're not needed. So not only do we free up valuable real estate on that server to introduce additional CPU and RAM, but we're actually eliminating a threat vector by eliminating potential vulnerabilities in those peripheral devices that you don't even need. We also extend this to our storage environment and our network environment. They are both custom and allow us to own not only the hardware, but the software and the firmware stack as well. And lastly, at a holistic level, our data centers have both logical and physical security. To that end, because we own the hardware, the software, and the firmware, we're not reliant upon any third party to provide patches in case of a vulnerability. As we control it, we can issue those patches as soon as possible. Now, you don't have to take our word for it. We do annual audits with our third-party consultants to test the attestation of our environment. And when it comes to genomics and the life sciences, that one certification that you may see on the bottom left is of utmost importance, and that's HIPAA. With Google, our entire cloud environment is HIPAA certified, which means you don't have to worry about selecting a certain cloud or instance to put that PHI data. As long as you put it on Google Cloud, it's automatically covered by our HIPAA certification. And you can see we have a vast amount of other certifications as well, such as PCI, FedRAMP, etc. And lastly, innovation is not about keeping all of it close to the chest. We are a firm proponent of the open source community. To that end, we've incubated numerous projects within Google that we've released to the open source environment. Two of those projects in particular, Kubernetes for container orchestration and TensorFlow for machine learning are the #1 and #2 highest engagement in all of GitHub. And not only do we incubate products and release them to the open source community, but our employees, our Googlers, also are active contributors. As of 2017, there were 1,100 projects in GitHub contributed by Googlers. And those Googlers totaled over 900 employees. So stay tuned for live Q&A. We'll be back in less than a minute.
All right, we're back.
All right, some questions from the audience. So I've been told this first question comes from a Jesus Gomez in New York who asks, do I have to bid for preemptible instances?
Yeah, the answer is no. This is a big difference with Google Cloud in that you don't have to bid for preemptible instances. You just select them. And Pipelines API provides an option for you to employ both preemptible CPUs and preemptible GPUs. You just use them, you get the discount, and you go to work.
And that's a flat 80% off for all instances, and GPUs is a flat 50%. So definitely take advantage of that. How do I get started with FireCloud?
Yeah, firecloud.org is the URL that you're looking for. And you can make an account. You actually get $250 in credits just to get started. So that's quite a few genomes, as you've heard about some of the really cool cost work that's being done by Broad. Yeah.
And, you know, I have a GCP instance. I know FireCloud runs on GCP. Do I have to create a new billing account, right? How do I get my, procurement team involved? Can I use my existing account with FireCloud?
You actually can. There's instructions on how to do this, both on our blog and on the FireCloud site. So you can bring your own GCP billing account. You can supply the billing account ID, and then the charge for all that compute goes to your home institution. So you're really getting all the benefits of FireCloud. You're getting all the benefits of the optimizations, and yet it's your home institution that's getting charged. So you don't have to worry about doing a transaction with Broad.
Excellent. Does the $5 genome include alignment, or is this just GATK? Great question.
Great question. So the answer is yes. So $5 gets you, yes, that's FastQ to VCF. It's the quality metrics, so it's alignment and variant calling. It's really exciting. While we're talking about secondary analysis, there's so much innovation happening in this field, especially from a cost perspective. We see GTK. It is really funny to think that it was really just a couple of years ago that it was 10 times more expensive, 9 to 10 times more expensive, and now we're talking about it. I think a lot of the buzz in our industry is always about the $1,000 genome and being able to generate the data cheaply, but bringing down the cost by 10x is pretty significant too on the informatics side. So I think that as we watch this field, we're going to see the cost of secondary analysis get closer and closer to zero, which is something I'm personally excited about because I think it unleashes all kinds of new research questions and opportunities.
And it sounds like we'll be at $3 very soon.
Exactly. Yeah.
Which I guess leads us to how would I get started with GATK 4?
Yeah.
Or GATK as a whole.
Yeah, exactly. So So again, if you want to run on the FireCloud platform, firecloud.org. Also, if you want to get started using GATK4, we've also put docs on our website. So if you go to cloud.google.com/genomics/GATK, you'll see docs there that help you just get up and running on a sample right away.
And I believe the Broad website also has.
And the Broad website has a
of,
similar URL as well,
GATK
and their blog. Yeah, so we both institutions have blogged about this. It's such an exciting development for the community.
And if all else fails, you can Google it.
Yes, we have a search engine to recommend too.
How would I get started with variant transforms?
So variant transforms, as you heard, I'm so excited about the work that's already being done with it. You're getting sort of a out of the box variant database based on BigQuery, which is such a powerful technology. So variant transforms, it's on our GitHub page. The URL is a little tricky, so probably Google that. But not only, if you go to the variant transforms GitHub site, you'll see not just the code, not just the documentation, you'll see our product roadmap. We've actually open sourced the product roadmap as well. You'll see the work that we're doing to add annotation support. You'll see the work that we're doing to add a BigQuery to VCF pipeline, because sometimes you might be selecting a cohort based on all the data you've merged and you want to go back to VCF. So you can use some file-based tools. So that's on the roadmap as well. Would really look forward to folks commenting on that. We really do look at that page, and it does inform our product development on variant transforms.
So another great example of contributing to the open source community.
Exactly. We realized that it was so important to make that open source. It's also written in Python, like many other stages of bioinformatics pipelines. And by open sourcing it, we know that folks will have their own VCF schemas. They'll want to be able to tweak it, and we want to just make that easier. And we're excited to give something back to the community in this way.
Awesome. Speaking of BigQuery, I've used it once or twice. And I see this thing about public data sets. Can you tell me which ones are available?
Yeah. Oh, man. I don't know if I can recite all of them, but gosh, there's a bunch. So some of the ones you might expect: 1,000 genomes, TCGA, the missing data set. There's an autism research data set. Is there some data from the Simons Foundation? There's a whole list online, and we'll make sure to post the URL afterwards so that you can find them. But we're always interested in public data sets that would be good for the community and that can be very helpful. So we see that as an opportunity, again, to aid the research community by hosting those data sets for free and then enabling folks to compute on that data set using some of the tools we've been talking about here today.
And speaking of tools, I know your team was at the HIMSS conference last week. Anything that you want to call out?
Sure, yeah. So HIMSS was a great opportunity for us to introduce the healthcare API. The healthcare API is actually built by our clinical data informatics team. Makes it really easy for you to deal with EMR and EHR data in the cloud. We're really interested in helping people manage that data, process it, and work with it at scale. So we're getting the signal to wrap up. So thank you so much for joining us. It's been a lot of fun, and thank you for all the great questions.